double ring on it. The adenine, same A that you see in ATP, same A is in the genetic code. And so there are two nucleotides, sugar phosphate base, joined together to make this hydrogen grabber. And so it grabs these two hydrogens. All right, so that's only going to work if and only if you have complex prerequisites like this hydrogen grabber and what? An enzyme that can use that hydrogen grabber. See, I could have introduced complex prerequisites earlier because to get this far, we've already had several enzymes, several great big protein molecules that can only be built using the energy that we're going to get from this process. So you have to have these molecules there to get the energy that will later on be able to make the molecules that make the energy that make the molecules that make the energy that make, you get the idea, okay? Well, here this NAD, we'll see a lot more of this guy later on, grabs these two hydrogens, and wow. Okay, what's left behind here is this molecule here. And I just, sometimes I just diagram the parts of the molecule that change. Now, this might not have grabbed your attention right away. But this is called a high energy, this is a really high energy bond. This particular kind of grouping here is called an anhydride without water. And it's a double acid. And so all of you know that acids and bases react. And of course, acids and acids don't usually react. That chemistry doesn't like that kind of combination. Acids and bases are supposed to react. And so in effect, by stealing these two hydrogens, this complex prerequisite turned a bland sugar aldehyde group into an acid group, and it was already attached to a phosphoric acid group. Now you've got a double acid. That'd be another way to say this. This would be a double acid, and that's not going to last long. And so this double acid, if you just left that in water for a few moments, it would break down. It would just disintegrate in water. Water would split it apart, leave the carbon acid behind the carboxylic acid, and the phosphoric acid would drift off in solution. And so you got to keep this reaction going quick, or water's going to break this apart. You're going to lose the phosphate you need to make ATP. And so what you've done is trick the molecule from being a low energy sugar group to a high energy double acid group, because you had the right enzyme and the right carrier in the right place at the right time, and you did it faster than water can break down this. By the way, speed's important to life too. Some of you've noticed that. You notice it more as you get older. There's times when I think I just can't breathe fast enough to stay alive, okay? I guess someday that may be finally true. Okay. So, here, this has got to happen real fast. You got this complex prerequisite, and it takes out this phosphate group and hands it over to, ADP. And so here's a kinase again. We name it a kinase regardless of whether it adds a phosphate or takes away a phosphate. And so here's a kinase that now takes away this high energy phosphate. This molecule here, the bond energy, and you can look these up in charts, is a lot more than ATP. And so the reaction can go from this molecule here to ATP. But ATP is an excellent example of what we just mentioned a moment ago, complex prerequisites. You kind of get the idea. Whoops. Well, let's be getting a bill for this projector here. Let's take it out of my salary, okay? To make ATP, a lot of times you get the idea, well, it's here you have a little phosphate, and whoo, here's ATP. No, you make ATP, adenosine triphosphate, out of ADP, adenosine diphosphate. You were already at almost all the molecule already there. And so here you had a double ring, a 6-carbon ring and a 6-carbon ring with 2 nitrogens in it, a 5-carbon ring with another 2 nitrogens attached to a 5-carbon ring sugar attached to 2 phosphates. So you had 90% of the molecule was already there, and all you're doing here is adding one more phosphate on the end. So you're not making ATP from scratch. You're making it from the Betty Crocker mix that has everything in it except add water and put in the microwave. Okay, so it's almost all there. So you're making ATP from another complex prerequisite, namely ADP. So you're not really starting from scratch, and it takes a lot to do that. But notice, ever since we split glucose, we've been carrying 2 molecules along. And so the 2 molecules of ATP that are made here are enough to balance the 2 ATP that we needed to activate glucose in the first place. So at this point, wow, whew, we broke even. Okay. Now, can you imagine how many millions of years, would billions, I guess, they usually think billions for the origin of life, would have to go on, until you could get past the break-even point. Now, how could you even get to the break-even point? You can only get to the break-even point with complex prerequisites, several great big protein enzymes, ADP and AD, and you got to have the glucose there to start with too, and so you got to have all this stuff just to break even. Okay, and you're trying to make a little energy, and all you've got is what? Back to where you started from, energy-wise. Now, by the way, that's not the end of the story. These molecules along the way, they are being broken down in my example here to make energy, but you can also harvest them for other uses in the living cell. And so in that sense, it's kind of like, plastics and oil. You can use oil or gasoline. Well, we'll take oil in general. You can break it down and make gasoline out of it to power your car, but what else can you do with it? You can make plastic out of it to build your car, okay, depending on the price you paid. Maybe mostly plastic. So plastic is both an energy, or hydrocarbons are both an energy source, gasoline, and a structural source, plastics. Well, that's the same thing here. Glucose is an energy source, but it's also a bodybuilder. And so the carbon skeletons are used to make fats and proteins and all of the other molecules, membranes in the living cell. But at least from an energy point of view, we're broken even. And look how much work we've done just to break even. Okay, we had to sneak on these extra 2 phosphates to get to the break-even point. Now where are we? And I call this the tricky phosphate sneak. Okay, and that's the part down here where this phosphate just out of the dirt you eat. Okay, so if you ever wonder, is there value in eating dirt? Yes. Okay, so here it is. And so dirt gets tricked into there and turned into high energy for you. And you wind up energetically even. The molecule that's left behind is now an acid. And it's still got a phosphate left in the third position, 3-phosphoglyceric acid. At the end of the split, yeah, that's here, isn't it? This is the way it looked a minute ago. Notice, well, that's not going to work too good, but maybe it won't work at all. But a moment ago, this was an HC double bond O at the top up here. So that's all it's changed, is that change from a sugar aldehyde group to an acid group.